So, dendritic cells will prime both CD4 and CD8 T cells. The CD4 T cells are absolutely crucial for helping the CD8 response. In order to get a full cytotoxic response, the CD4 is providing stimulation to the CD8. The CD4 T helper cells are also required to give an antibody response, a mature antibody response, which includes IgG. And IgG is what you want in the vaccination. In order for the T cells to help the B cell, they need to meet, and they do this in the follicle. If you look at the top of this slide, there is a picture of a lymph node from a monkey. We are interested in the follicle. The follicle is also called the germinal center. This is where the T cells and the B cells interact, and the T cells that go in here, they upregulate a molecule on their surface called CXCR5, which helps them locate into the germinal center. So, naive CD4 T cells, which are predestined to become T follicular helper cells, they need two things. Firstly, they are innately producing more IL-2 as in their naive state, and this is a determinant factor for becoming a T follicular helper cells. They also need stimulation from the ICOS ligand over their ICOS, so they need to express the molecule ICOS. The crucial step in the development to becoming a T follicular helper cell is regulating BCL6, which is a transcriptional repressor. BCL6 expression makes it possible for the CD4 T cell to upregulate CCR5 and then migrate into the lymph node germinal center, which also exists in the spleen. Inside of these germinal centers, the T follicular helper cell is meeting the B cell, and the priming from a second antigen presenting cell, which is most commonly the B cell, will complete the last step of the maturation of the T follicular helper cell, which is a type of effector cell. Here is a recent image illustrating the function of the T follicular helper cell by Shane Crotty, which is very important in this field. So first, up to the left in the input site, we see the dendritic cells, but also macrophages and monocytes that can work as antigen-presenting cells and provide co-stimulation cytokines for the activation of the T cell response. Activated B cells on the very top also provide co-stimulation and antigen presentation, while they in turn receive help from the follicular T helper cell. The germinal center B cells, which is about further inside of the germinal center, will help the differentiation of the T follicular helper cells. The follicular dendritic cells are very rare cells, and they are present in the B cell follicles. They store and present antigen to the B cells. The function of the follicular dendritic cells is not entirely clear when it comes to T follicular helper cells. Another T follicular helper cell is the T follicular regulatory cell. This regulatory cell will prevent activation and function of the T follicular helper cell by the inhibitory receptors PD1, CTLA4, and TIGIT. So the effect of the T follicular helper cell on the B cell response is the providing of IL-21. And this is very important for the activation and the differentiation of the B cell. The B cell must differentiate into plasmablasts and plasma cells in order to provide IgG. IL-4 is produced at the later stage of the T follicular help provided to the B cells. If the germinal center B cell does not get the help it needs, it will not survive and it will cause death. However, if the T follicular help is provided to the B cell, it will proliferate inside of the germinal center. And then in this way, there will be more plasma cells produced. This will also lead to B cell memory, which is a very desired, highly desired feature in the vaccination. This proliferation, stimulation for proliferation, is given from the T follicular helper cells by a co-stimulation with the CD40 ligand, which is important for the differentiation and the survival of the B cell. So now we have identified several points in the vaccination where it must work. And the first is to use an immunogenic antigen, and this is what we will come to next. The DC activation we have been through, the B cell stimulation and the development of the T follicular helper cells, and basically using the correct type of helper epitopes, which stimulates an effective B cell response. We talked about that. The T cell stimulation and differentiation is true for both the CD4 and the CD8. And here we also need a T cell memory that we can detect. So the basis for the long-term protection is the B cell memory and the T cell memory. And there are reasons for why a vaccine would fail to induce these memories, and we will try to go into that later on. So all vaccine design is requiring two absolutely crucial steps, and the first is the antigen design. What antigen from the pathogen to deliver, and also how this structure is supposed to look like. Second part is the adjuvant, which we have mentioned is really important for stimulating the dendritic cell and the innate immune signals. The antigen design is must be performed in a way so that the antibody response is directed towards regions of the pathogen where it will prevent the pathogen from infecting. And this is primarily true when it comes to virus. It's not as easy to neutralize the bacterial binding and entry. So for antiviral vaccines, the absolutely most desirable antibody response is an antibody which binds to the virus to prevent it from binding or entering cells. And in order to achieve this, to get antibodies which are directed towards these epitopes, the neutralizing epitopes, we need to deliver the antigen in a native folding. And this is to provide the structures that are constituting the B cell epitopes. So B cell are not only recognizing sequence, they also recognize structure. And some B cell epitopes may even be from two different parts of the protein. In addition to maintain the integrity of the antigen, we must make sure that we do not just include the small part of the antigen which gives rise to the neutralizing antibodies. It's also important here to include the T cell epitopes, which are giving a good T cell response in the natural infection. And in order for the T cells to provide help to the B cells, which recognizes the same protein, the T cell epitopes must be present in the same vaccination. And although there are some T cell epitopes for T help, which seem to be general, which seem to stimulate all of the HLA, there are considerations because different individuals have different HLA and would also then present different epitopes. So therefore, we must provide an antigen which is large enough that it can contain epitopes which all the different HLA can present. Now, here we go to the adjuvant. The adjuvant is providing signal 2 and signal 3. So signal 1 is the presented peptide. The signal 2 is the co-stimulus, and this is the upregulation of the B7 molecule CD80 and CD86 and also CD40 on the surface of the dendritic cell. The adjuvant should also induce cytokine production, which is signal 3. The cytokine production will not only be a prerequisite for the priming, it will also enhance the effect on the innate immune system. So the cytokines will also other innate immune cells. The adjuvant should not be too toxic. All adjuvants are toxic to some degree because the destruction of tissue is an important danger signal in itself, and it's hard to have an adjuvant which is completely non-toxic. And also important with the adjuvant is that it should stimulate the right type of response. And so what is the right type of response? We will come back to that. But primarily for viral infections, especially, primarily a type 1 response. And I think at this we will come back to the types of the response later on and discuss that. What you also would like to do is some kind of broadness in the type of response that you get, but you would like to avoid having a primarily type 2 response, which is what you get with, for example, an adjuvant like alum. So these are the two most important points, the antigen design and the adjuvant. So it is commonly thought that you should try to make your vaccination mimic the natural infection of the pathogen against which you would like to vaccinate. This means that vaccination against virus and intracellular bacteria may mainly produce a Th1 response. The Th1 response is a very effective way of inducing T cells, especially then cytotoxic T cells. And the interferon gamma, which is produced by the cytotoxic T cells and from the Th1 helper cells, will also help macrophages to destroy intracellular bacteria. So when we're dealing with virus, the Th1 type of response is the most required response. The Th17 response is analogous to the ILC3 response, which we talked about before, and here it's mainly a neutrophil type of response, and this is not necessarily desired, while neutrophils can cause a lot of tissue damage. When it comes to a Th2 response, we do not vaccinate against helminths, so against parasitic worms, but triggering a slight Th2 response in addition to the Th1 response is not necessarily a bad thing, while the Th2 response is very efficient at stimulating antibody production, since the IL-4 and IL-5 are both required for the later steps in the antibody production. So all different helper cells can support the antibody production. But by combining a Th1 and a Th2 stimulation, then the effect on the antibodies is enhanced. Now, we would, to all effect, want to avoid a pure Th2 response in a vaccination. And why is that? That if we then get challenged with the virus or the bacteria, firstly, it's not very effective for the protection, since we don't have a strong induction of cytotoxic T cells, but also there is a risk for inducing an allergic response. And particularly, this is not desired when it comes to pathogens which affect the lung. So we want a Th1 response, and this is a kind of response we will get from TLR stimulation.